We are seeing an explosion of these strategies, especially in Silicon Valley right now, with multiple types of sensors that live on your body. You may be familiar with the Apple Watch and Stanford's projects on the heart. Those are leading to prediction of who is going to be at risk and who is having an unknown arrhythmia of the heart, for example. The Apple Heart Study, as it's called, is changing fundamentally, taking care of individuals that we would have had a much more expensive strategy for doing that same care. Similarly, Google and the contact lens strategies are leading to electronic contact lenses that are in fact able to measure glucose levels in your tears and relate those glucose levels in your tears to your blood. And a large trial with Novartis is looking at diabetic patients that way. In my own labs, we've been developing a smart bra, which constantly measures breast health and looks for earliest changes in breast tissue to be able to put together breast cancer detection at an early stage. These things seem like they're far off, but they're not far off. They're evolving very rapidly, and we're having the luxury of being able to evaluate some of these in early clinical trials in the Bay Area. We believe, though, that your home offers much better opportunities. People that wear devices on their body don't tend to keep them on. The average time someone puts a Fitbit on is about 6 to 9 months and then stops wearing it. The best way to monitor a human is in a way in which they don't have to change their behavior. If they can go about doing their normal business and not have to do something different, and yet you can monitor their health state repeatedly, serially over time, then you have a much better opportunity to intervene at an earlier point in time. We think the bathroom will be one of the most critical components of this. And over the last several years, we've been building smart toilet technology, and we will introduce this later this year in clinical trials. In this smart toilet, which can be adapted to any existing toilet, and the eventual cost of under $100, the goal is to do simple, ultrasensitive, multiplex biomarker detection. This means that one will be able to measure biomarkers in your urine, in your stool, detect the kinds of diseases that are shown here on this slide. At the core of this are technologies like the magneto nanosensor shown on the right that is able to do very high precision detection of biomarkers in bodily fluids and multiplex those across a large number of biomarkers. The world of in vitro diagnostics is rapidly growing. What you'll see is that we're able to, in fact, mine the blood in a much deeper way as a global community than we've ever been able to. I think this is one of the biggest threats, in fact, to many of the current nuclear medicine procedures and radiological procedures, and I'll give concrete examples in a few minutes. But we're seeing a lot of new technologies that are allowing us to mine, especially circulating tumor cells and microvesicles and exosomes in your blood. Because these give us a direct insight and window into the tumor, for example, they let us, without a solid tumor biopsy and instead a liquid biopsy of the blood, to determine what's going on at the tumor site. Something that has typically been in the realm of imaging is shifting to where you can measure many changes in biomarkers by not having to image the tumor directly as we do in molecular imaging. We're also asking questions about what can we do to start to force the body to make biomarkers it otherwise would not make. Could you develop a pill that would allow you to know if there's a cancer hidden somewhere in your body by forcing that cancer to secrete a biomarker it otherwise would not have made? In this approach, we use small segments of oligonucleotides, in this case called mini-circles, that are driven by that pink switch that you're seeing on the blue circle. These mini-circles enter your body and go to all cells within your body. So the mini-circles are not targeted in any way in the current incarnation of this work. The mini-circles are able to circulate throughout your body and then only activate when they encounter the environment of a tumor cell. So in this case, if you have a lung cancer, relatively small, let's say even at just a few millimeters in diameter, and we look at the blood supply to both tumor cells and normal cells in the vicinity of that lung cancer, what you can see are the mini circles going out into normal cells and tumor cells. But when they are in a tumor cell, a specific promoter, that pink switch, activates, leading to the production of a biomarker shown as that green rod. That biomarker then enters the blood, and in our case is able to be sampled either from the blood or now in the urine, and in a new paper coming out in the breath, telling us that there is a cancer hidden somewhere in that person's body. That then allows us to also image up to a certain length of the signal, a certain degree of the signal within the body, and also lets us treat the tumor. These kinds of approaches are rapidly evolving, where we're not dependent on what nature provides as a biomarker, but instead are pushing what we call synthetic biomarkers to force tumor cells to do something they otherwise would not do. That leads us to precision medicine, which is once you have these approaches, you still need to, in many cases, treat very precisely based on the genetics of that particular tumor and on the biology of that particular person and tumor. And in this area, we really see in vitro diagnostic strategies rapidly growing. And this will challenge the role of molecular imaging in patient management. We know that molecular classification is predominated by biopsies with their limitations, blood samples with their limitations, and traditional imaging. And there is a huge opportunity for molecular imaging in this area because of the limitations of each of these approaches. Nevertheless, the low costs, especially of sampling the blood, are leading to disruptive ways to monitor precision medicine at work. For example, if we focus on EGFR, epidermal growth factor receptor, the traditional way might have been to biopsy the tumor and try to determine mutation status of that tumor with respect to EGFR. But one can, in fact, sample the blood instead and determine a lot about what's going on at the tumor site. In this work, we take blood from you, and in that blood sample, we separate out the circulating tumor cells from white blood cells and other cells using a magnetic sifter. Magnetic nanoparticles bind to only predominantly the circulating tumor cells. That lets us sift the circulating tumor cells magnetically, and then we can sort each circulating tumor cell into a nanowell and interrogate each of your circulating tumor cells for what genes are mutated in that cell relative to other neighboring cells. This then allows us to fully diagnose mutational evolution in a tumor by looking only at blood samples from a person before start of therapy and during therapy. This approach totally circumvents the need for imaging and raises the question, could eventually tools like this be used instead of serial imaging to monitor response for therapies and potentially even to predict response for therapies? In a few minutes, I'll show you another way that we're doing this with a new PET tracer. But the key here is that we as a community have to be very well aware that in vitro technologies are very rapidly growing. And instead of serially imaging patients, it could be that one might image the patient much less frequently, and in the interim sampling sites, only sample blood. So how can nuclear medicine and molecular imaging position itself to be a key player in precision health and then also evolve in precision medicine? Well, it can potentially lead the way in detection and staging after in vitro diagnostics warn of a potential problem. Whether it's the smart toilet saying there's a potential problem, whether it's a blood sample, maybe molecular imaging is that next cost-effective step to determining the site of that problem. We are seeing a huge explosion of regional molecular imaging technologies that are able to more sensitively do what we can currently do in nuclear medicine at the whole body level. Nothing beats SPECT and PET when you want to interrogate the entire body. But if you're willing to take a trade-off and only image a region of the body, then new tools start to open up that allow you to, in fact, get a greater sensitivity and the potential for detecting very early, in this case, cancers. I don't have time to show all of these, but I'll focus on one recent one, which is molecular ultrasound. In this approach, a gas-filled bubble is, in fact, used with targeting agents on its surface to target vascular endothelium that's feeding tumors, including early tumors. Because the gas-filled bubble is responsive to ultrasound, you yell at it and it yells back. The beauty of this is that you don't have to use a radioactive agent, you have a long shelf life, and you have a ubiquitous technology like ultrasound. So in this work, after many years in working with the US FDA and BRCA, we were able to take these bubbles into human applications. And this just shows one example of a 59-year-old female with serous cystadenocarcinoma. And in panel A is the traditional ultrasound image with transvaginal ultrasound. Panel B is microbubbles that have locked into the tumor 15 minutes after intravenous injection of the microbubbles. And C and D are just histological proof of blood vessels and the presence of the target on the blood vessels, which in this case is KDR. These are currently leading to about 85 to 88% accuracy between the imaging study and histopathology. And these are just in their first generation, and we think they will rapidly improve. So these kinds of technologies give us some promise that as in vitro technologies take off, molecular imaging will have counterparts that may be able to detect very small lesions, in this case in humans currently down to about 3 millimeters, that will in fact enhance our ability to be a part of the precision health strategy. Of course, PET may also play a significant role here. In this work, this is a Naughton peptide that we've been engineering for several years, and in a clinical trial that's going on at Stanford and at Peking Union Hospital, we've been taking a gallium-labeled version of this peptide and a fluorine-18 labeled version of this peptide. This peptide binds alpha V beta 6, not to be confused with alpha V beta 3. This particular biomarker is highly upregulated in specific cancer types, including ovarian cancer, but our target predominantly is pancreatic cancer. And it's also highly upregulated in cervical cancer, and it's not present in pancreatitis and other confounding disease states. In this first in-man image that we obtained just a few months ago, you're looking at a pancreatic cancer that is in fact being lit up because of homing of this peptide to the head, the neck, and the tail of this pancreatic cancer. These kinds of approaches give us hope that there will be more such approaches, just like as we're seeing in prostate cancer imaging, where FDG is not able to do a sufficiently robust job. When it comes to pancreatic cancer, ovarian cancer, and a few others, we hope that we'll continue as a field to see more imaging agents that will be useful in these approaches. Nuclear medicine can also lead the way in predicting and monitoring response to therapies. Just as in vitro diagnostics is challenging us here, we do have the ability to develop many new approaches to predicting and monitoring response to therapies and have the advantage of spatial information that cannot be provided by in vitro diagnostics. In this recent work just published a few weeks ago, we've taken a small molecule that is able to bind specifically to mutant EGFR. So we have the epidermal growth factor receptor, but then in many lung cancer patients, mutant epidermal growth factor receptor. And in this work, we're able to, in fact, build a tracer that is binding mutant EGFR, and then ask the question in a patient, is their tumor likely going to be responsive to a tyrosine kinase inhibitor? We can therefore take a set of patients and begin to stratify them. who should get the tyrosine kinase inhibitor and who should go on to other treatment methods by doing essentially that virtual biopsy entirely through molecular imaging with PET. In this work, what is done in approximately 100 patients is that we've taken that particular molecule, 18F-MPG, and if you look at the top row, that is one patient with a mutation in EGFR, and you'll see their CT scan, then their FDG scan, and then their MPG scan. And you'll see that in all three, you see tracer uptake here. You see in the FDG and combined FDG PET CT, and then in the MPG and combined MPG PET CT uptake of this small molecule tracer, indicating that you are binding to a mutant EGFR. You go to the middle row, and that's the second patient who also has a mutation in EGFR, but a different kind of mutation, in this case a point mutation. And again, the tracer is taken up nicely in both the FDG study and in the MPG study. But when you get down to a wild-type tumor, you of course have uptake with the FDG scan, but not with the MPG, because the MPG tracer does not bind to the wild-type EGFR. This nicely lets you determine, totally non-invasively and in a spatially informative way, the mutational status before you begin therapy and actually as you look at the evolution of that therapy. This then leads the ability to have truly an analysis of who's going to benefit from a tyrosine kinase inhibitor well in advance of actually using that inhibitor. So on the left graph are the results by actually looking at the progression-free survival in time is a little over a year, but by looking at the mutational status directly. And on the right is by looking at the 18F-MPG tracer uptake with a cutoff in SUV of around 2.2. And what you nicely see in this approximately 100-patient study is the ability to stratify who is going to have progression-free survival with the 95% confidence intervals shown as the dotted lines versus who is not, thereby letting you a priori determine who are the individuals that you should be selecting for the therapy. Something that you might argue you could again do with an in vitro diagnostic and circulating tumor cells, as I showed earlier, but do perhaps in a way that's better using spatially informative information and not just of the primary tumor, but of any metastatic sites. Nuclear medicine will also lead the way in imaging of the immune system. We've heard great talks at this meeting, including how we will use nuclear medicine to monitor many immunotherapies for cancer. Just as we can measure small molecules like tyrosine kinase inhibitors, we can do very complex analyses of immunotherapies. There are many such targets that are evolving in the immunotherapy world: checkpoint inhibitors, different molecules that interact with the immune cells and immune cell tumor cell interactions, and then immune cells themselves given as the therapy, such as in CAR T-cell therapy. We've been for many years developing reporter gene technologies for use in immunotherapies. We originally started the work for gene therapies, but then we're able to evolve them for immunotherapies. In this case, cells are labeled with a reporter gene so that every daughter cell carries that reporter gene. And so now, when you move towards imaging of CAR T cells, one can take cytolytic T cells, and as you modify those T cells to do a better job in homing in on glioblastoma in this case, you also insert a PET reporter gene. That leads to the ability to track those cells for the rest of their lives. Unlike radiolabeling them with indium-oxine or a radioactive agent, because they're genetically barcoded, you can monitor them forever in that human. In this work, which took almost a decade between the City of Hope, UCLA, and Stanford, we were able to demonstrate for the first time in humans the ability to do this. Here you look at one particular patient example in which on the left is a tracer that will home in on the PET reporter gene labeled cells. And before we give the cells, you see very low signal from the superimposed PET-MR images. But after we give these CAR T cells and then give the PET tracer, you see nice homing of this tracer to only those areas that have significant number of CAR T cells. This lets us directly answer questions like, why are the CAR T cell therapies failing? Why is it that cells are not potentially reaching their target sites, in some cases due to death of the cells? What is happening in terms of the engagement of the cells with the tumor? And as you'll see in a minute, can the cells actually find sites of tumor within the brain that are otherwise not known to us? There is a huge opportunity in immunotherapy for nuclear medicine to make these therapies achieve the potential they have. Otherwise, the field will continue to shoot blindly, and we will not achieve the eventual outcome that we may achieve by simply, in fact, following the cells and the molecular processes at the molecular level. These approaches are rapidly growing. We have many trials going on now with CAR T-cell therapy in which PET reporter genes are being incorporated, letting us follow those cells over long periods of time. This just shows you one example in which the CAR T-cells not only home to a tumor that we knew about, the site on the left, but also went to a second site that we did not know about. So there is a huge opportunity in precision health to, in fact, be more proactive. For the younger people in the audience, I believe the future is not about just doing what we've done, which is being very focused on therapies, very focused on late-stage diseases. Although I've used cancer as an example, the real potential lies in being much more proactive. How do we develop the next generation of technologies that will prevent disease, monitor you in the home, monitor you all the time so that we can intervene at the earliest possible time when we have the greatest chance for success in achieving a healthy life outcome. To end here, in the last 2 minutes, we've initiated a very large project called the Baseline Project in combination with Google and Google Life Sciences, known as Verily, and Duke University. We are doing one of the most extensive studies of human health ever attempted. The goal is that we, again, look at people too late. We look at people after they present with symptoms and after they already have disease. What we really need to do is to study people over many years, long before they become ill. In the Baseline Project, with over 10,000 subjects initially, and hopefully to grow to many more, we do the deepest possible analysis by looking at all aspects of what we can measure: their saliva, their tears, their breath, their urine, their stool, the microbiome, the full genome, full molecular imaging. And we do this serially along with wearable devices to look for their health state. We believe these kinds of studies will fundamentally shed new insights and will be the Framingham studies of the future. This is how we will learn to truly prevent and then intervene early in disease. This study, which is at a cost currently of almost $800 million, will soon go over $1 billion. And that's why it's not been able to be attempted before. But we believe studies like this are what will be critical to achieving a precision health outcome. So in conclusion, I've tried to give you a glimpse of some of the ways we're thinking about precision health, which is different than precision medicine. We hope precision health strategies will help move the entire field of healthcare to be more proactive. In vitro diagnostics will likely play a greater role and may decrease the utilization of molecular imaging. So we are working hard to be a part of that instead of watching it happen from the sidelines. I have recently started to work with the chair of the Department of Pathology at Stanford to bring the two departments, radiology and pathology, into one integrated department. We believe radiology and pathology need to be consolidated, and that's what will occur over the next decade. Regional molecular imaging technology will likely play a greater role in early cancer detection. I showed you examples of the targeted microbubbles. Many approaches are evolving that will allow molecular imaging and hopefully nuclear medicine with higher sensitivity to go after smaller and smaller volume lesions. And finally, this does not mean that there isn't a great role for nuclear medicine. I showed you examples in cancer staging with alpha V beta 6 imaging of pancreatic cancer, patient stratification with 18F MPG for determining epidermal growth factor receptor status, and a new approach with PET reporter genes for monitoring CAR T-cell therapy. So all of this is made possible by the work of many people. I'm privileged and honored to lead a great group of scientists at Stanford and the entire Department of Radiology, and also special thanks to Dr. Andrea Garu as the head of nuclear medicine at Stanford. And we hope that through the global community effort that there will be a huge, bright new future coming for all of precision health, and that nuclear medicine will play a great role for it. I also want to especially thank our colleagues in China at Harbin Medical University and Peking Union Medical College. And special thanks to you for allowing me to present this vision. Thank you very much.
Thank you for showing the expected features of the future. And we now feel the challenges and opportunities, how to set our nuclear medicine role in the future. And in our present state, I'd like to give him the appreciation plaque and give him a great hands, please.